Hi, good morning, everyone. Good morning, ma'am. Good morning, classmates. For today, we'll discuss about lipids and lipoproteins. This morning, we'll be tackling about exogenous pathway of lipid metabolism, the endogenous pathway of lipid metabolism, and atherosclerosis formation. Hello everyone. For our first topic, we'll be tackling about exogenous pathway. In exogenous pathway, we have diet, right? It gives character in green, gives triglyceride in cholesterol. So the triglyceride or glyceride and cholesterol are absorbed. And then they absorb in the intestine, specifically in the small intestine. In the intestine, the triglyceride and cholesterol becomes chylomicrons. Chylomicrons, these are samples of lipoproteins. Now the chylomicrons enters the lymph, and then they are responsible for white cloudy and sort of buttered component of the serum after you have been taken a fatty meal. And then chylomicrons is a water insoluble, so which it cannot enter the circulation, thus pupunta siya sa lymph. And then in the lymph, mag-drain siya into thoracic duct. And from thoracic duct, it will go down through the bloodstream. So now in the bloodstream, your chylomicrons will have its own apolipoprotein, which it helps to circulate the circulation. So the apolipoprotein are smaller lipoprotein that would help in the circulation of our water-insoluble lipids. So for the chylomicrons, the apolipoproteins here is the B48. I mean B48. So the chylomicron, once it enters the circulation, it will be bound to our B48, and your B48 is an apolipoprotein. Now, since it is a apolipoprotein, it will activate the LPL that is seen in the vascular endothelium. So, what is LPL? That is your lipoprotein lipase. So, LPL is basically a lipoprotein. So, as continuation, the lipase lipoprotein is an enzyme that is responsible for esterification of fats. So, when you say esterification, um, it means that it is a acidification, or is the process of fat being metabolized into smaller components to use as raw materials either for energy production or raw material for storage. So the apple bean will activate and it will interact with your LPL. And after sila nag-interact, it will cause its activation, so that the apple bee activates your LPL. And when the LPL is activated, there will be esterification na mangyayari. So what happens when there is esterification? There is a cleavage or breaking down. So when it breaks down, the chylomicrons will form cleavage, and the cleavage of chylomicrons will produce a fatty acids. So the LPL is activated by B48, attached to chylomicrons, and it will initiate the acidification of your LPL to your chylomicrons. So in the other words, cleavage or breaking down of your chylomicrons to produce fatty acids. So we have here the fatty acids. So the fatty acid has two paths. So number one, the most of the fatty acids will be given to muscle cells to be used as a fuel in order to provide or to supply the energy demand of the body, specifically the muscles. And then very small fractions of fatty acid that will remain. And it will be bound together by glycerol to form triglyceride. And it will be deposited into the adipose tissue. So lastly, the remnants that will remain after the cleavage of chylomicrons will be given to the liver to be part of your bile and again plays a vital role in emulsifying fats during the lipid digestion process.
So, a brief review about the endogenous pathway. So, in endogenous pathway, we are dealing with lipids that are already stored in our body. So, the liver, the liver will use a fatty acids in order to form or synthesize triglycerides and VLDL. So the nasal HDL will gather LDL that are forming the sarcoma. Fourth, the TL3 will undergo esterification promoted by your lectin cholesterol acid transferase and will form its esterified form called HDL2. So next is triglycerides. So the triglycerides, it is a storage form or it will be stored in your adipose tissue. So and your VLDL, since its lipoprotein has its subsequent apolipoprotein that will help it circulate. So next is the three apolipoproteins will activate the lipoprotein lipase present in the vascular endothelium in order to convert your VLDL to become IDL. And lastly, LDL may be used to synthesize all cell membranes in the form of phospholipids like glycolipids. So that's all. So the third one is the atherosclerosis formation. So first, we will review the topic that we have discussed last time. So 30 of the form LDL will be used in the cell membrane as cytogenesis, and the excess LDL from that 30% will block the vascular endothelium of big arteries in the body, and that will lead into what we call as atheroma. So first, we will define what is atheroma. So atheroma, it is a term or it is the term used to define gaseous material containing high amounts of lipids. So it is found in plaque-like thickenings of the interior portion of the vessel wall. So normally, if the atheroma formed is only a small blockage excess LDL, then the HDL would just get rid of those LDLs. So however, in cases in either first of having a low LDL or a good cholesterol or number, or second, having a high value of bad cholesterol or LDL, then the atheroma would become something that cannot be dealt with effectively. The HDL and the atheroma formed would lead into what we call atherosclerosis.
So, first, we will define what is atherosclerosis. So, atherosclerosis, it is a disease in which plaque builds up inside your arteries. So, plaque is made up of fat, cholesterol, calcium, and other substances found in the blood. So, over time, um, plaque hardens and narrows your arteries. So this limits the flow of oxygen-rich blood to your organs and other parts of the body. So atherosclerosis can lead to serious problems including heart attack, stroke, or even death.
So the next one we'll discuss about how atherosclerosis formed. So atherosclerosis is there a blockage that would form inside the vascular endothelium of large arteries. So atherosclerosis, it is a condition wherein the lesions would form primarily in the tunica intima of large elastic arteries in your aorta or human arteries surrounding the heart. So these lesions, it would develop following endothelial injury, so that the endothelial injury would lead into endothelial dysfunctions or functioning. So these are said to be the most acquired abnormality of blood vessels. So the complications of atherosclerotic disease would include the first, the ischemic heart disease; second, the myocardial infection or heart attack; third, stroke; fourth, gangrene of the limbs.
So next is we will tackle about the factors that predispose to endothelial injury would result in the formation of atherosclerotic lesions. So first is the LDL cholesterol hyperlipidemia. So it means that it has a high lipid concentrations. So high, it means that it has a bad cholesterol and good cholesterol, resulting into formations of atherosclerotic lesions. Second is hyperglycemia, specifically in diabetic patients. So most of our fatty acids and triglycerides are formed from lipogenesis, which usually happens where there's excess in glucose in the body. So third is the hypertensions. Hypertension. So hypertension, the pressure of the blood. So in hypertension, the force induced by the blood when it bounces course that would result into vascular injury would occur. Fourth is the increased toxins level associated with cigarette smoking and certain bacterial and viral infections. So in this would cause vascular injury for bacterial infections. It would cause chlamydia, pneumonia. For viral infection, CMV or cytomegalovirus. So these two are associated with targeting vascular endothelium, resulting into endothelial injury that will lead you to formation of your atherosclerosis or atherosclerotic lesions. So next we will tackle about this diagram. So how atheroscleroma or atherosclerotic lesions would form inside the vascular endothelium. So first factor is that the altered function. So in vascular injury as a result of LDL or as a result of high LDL concentration, atheroma forms in our blood vessels. If ever that there has a decrease of concentration, good cholesterol, the body would get rid of the LDL from vascular endothelium. So it will cause increased permeability. When there is an increased permeability in our LDL, cholesterol will now enter to mica and it will allow the entry of the LDL from the vascular endothelium layer. So the one single layer of squamous epithelium, this would result in increased permeability to LDL cholesterol, would increase the adhesion of the WBC into the endothelium. So that's all. Thank you.